Dear students, now I am going to talk you about gamete transfer, fertilization event and post-fertilization events. We have already learned that there are three stages of events of sexual reproduction. They are pre-fertilization events, fertilization event and post-fertilization events. In pre-fertilization events, there are two sub-stages, right? They are gametogenesis and the transfer of the male gametes. We know about gametogenesis. Now let me tell you about gamete transfer. Transfer of male gametes to the female gamete is called gamete transfer. You know, if this process is failed, there will be no fertilization, isn't it? So gamete transfer should be surely done for fertilization, right? In several simple plants like algae, bryophytes and pteridophytes, water is the medium of gamete transfer. You know, a large number of male gametes fail to reach the female gametes. To compensate this loss of male gametes during transport, the number of male gametes produced is several thousand times the number of the female gametes produced. You know, in a majority of organisms, male gamete is motile and female gamete is stationary. Exceptions are a few fungi and algae, in which both types of gametes are motile. In flowering plants, you know, pollen grains are carriers of the male gametes, and male gametes are non-motile in flowering plants. So pollination facilitates transfer of pollen grains to the stigma, right? You know, this picture is pollen tubes. Okay, these pollen tubes carry male gametes, right? Now let us know about fertilization. As you know, fertilization is a fusion of male and female gamete to form zygote. This process is also called syngamy. Here, let us try to know the differences between fertilization and syngamy. Now let us know the types of fertilization. Depending upon the place of syngamy, fertilization is of two types, external and internal. External fertilization is the type of fertilization in which the fusion or syngamy occurs outside the body of organisms, right? It is very common in most algae, some fungi, several invertebrates, bony fishes and amphibians like frogs. Organisms exhibiting external fertilization show great synchrony between the sexes, male and female sexes, and they release a large number of gametes into the surrounding medium to enhance the chances of syngamy. You know, there is a major disadvantage in external fertilization. The major disadvantage of external fertilization is that the offspring are extremely vulnerable to predators, threatening their survival up to adulthood. Now let us see internal fertilization. Internal fertilization is the type of fertilization in which the fusion or syngamy occurs inside the body of organism. It is very common in terrestrial organisms, but some aquatic and semi-aquatic organisms also perform internal fertilization. In this event, though the number of sperms produced is very large, there is a significant reduction in the number of eggs produced. Now, parthenogenesis. It is a special type of reproduction in which the female gamete directly develops a new individual. Parthenogenesis occurs in honeybee. All the individuals produced by parthenogenesis are haploid because they are directly developed from the haploid female gametes. Understood? You know, in honeybee, both fertilization as well as parthenogenesis occur. If parthenogenesis occur in honeybee, drones are produced. So drones are haploid because they are developed from haploid gametes, haploid female gametes. If fertilization occur in honeybee, female honeybees are developed. So females are always diploid. Yeah? Post-fertilization events. We all sexually reproducing organisms are started from a single cell called zygote. Development of zygote depends on types of life cycle of the organism. Secondly, the environment in which it is exposed to, right? You know, in fungi and the algae, the zygote before germination undergoes a resting period. A thick wall is developed around it, which prevents it from desiccation and damage. In organisms with haplontic life cycle, zygote divides by meiosis to form haploid spores, which grows into haploid individuals. Right? Now, after zygote formation, we have embryogenesis. Process of development of embryo from the zygote is called embryogenesis. If the organism is in haplontic life cycle, the zygote will undergo meiosis and spores will be formed. Spores will undergo mitotic division and develop as embryo. Right? So, if the organisms are having the haplontic life cycle, the zygote will undergo mitosis and the embryos will be developed. Understood? Now, let us see different types of animals based on whether the development of the zygote takes place outside or inside the body of the female. Organisms are of two types. If the basis is given in this condition, they are oviparous and viviparous. You know, you have already learned that the animals in which development of zygote takes place outside the female parent are called oviparous. These animals lay fertilized eggs covered with hard calcareous shell in a safe place in the environment. Examples of oviparous are reptiles and birds. You know, viviparous. The animals in which the development of zygote into a young one take place inside the body of the female parent are called viviparous. Right? You have understood already. After attaining a certain stage of growth, the young ones are delivered out of the body of the female organism. Right? The chance of survival of young ones are greater in viviparous organisms because of the proper embryonic care and protection. Understood? These are oviparous animals. These are viviparous animals. Now, let us know about the embryogenesis in plants. In flowering plants, the zygote is formed inside the ovule. After fertilization, the sepals, petals, and the stamens of the flower wither and fall off. The zygote develops into the embryo, and the ovules develop into the seed. The ovary develops into the fruit, which develops a thick wall called pericarp that is protective in function. Look here, endocarp, epicarp, mesocarp. These are parts of pericarp. They are developed from ovarial walls. This seed is developed from ovule. Right? Okay, let's learn this much only for today. In next class, we will have new chapter. Have a nice day.